All right, welcome everybody to the latest installment of Apiculture Online, Hive Chat with NC State. Appreciate everybody attending this evening. We were just talking offline earlier here about how this is one of the real difficult times of the year for beekeepers because it's so blessed hot and the bees still need a lot of attention. It's just hard to kind of get up and muster up the energy to be able to do that, but hopefully will inspire you this evening to really get the energy to be able to do that because your bees definitely need you right now. We were also talking about strategies to do that. One of the best strategies is to do things really, really early in the morning before the heat of the day. You'll like it, and the bees will probably like that too. But I'm going to go ahead and share my screen. And go ahead and get started here. We have a really great lineup today. We have our postdoc located at UNC Greensboro, Dr. Ishmael Amiri, who's going to be talking about some of his research, and then his co-advisor, who is still has an appointment at UNCG, but is physically located in Canada right now, in Alberta, in perhaps a transition to a new position up there, Dr. Olav Ruppel. And so we're really lucky to have him on the chat tonight as well. But as usual, we're going to start out with the first segment talking about bees in season, and that is what your bees are doing right now and what you should be doing as a beekeeper. And if you haven't noticed, it's pretty hot out. And I think this is true probably all the way up your way too, Olav, right? I mean, the jet stream is so darn high. You guys might be in the 90s as well. But certainly down here, it's been hot and wet. So summer has finally arrived and with a vengeance. Hot and humid is definitely a recipe for growing parasites and pests, especially varroa mites and small hive beetle. They really prefer these kind of warm and wet conditions, especially the high humidity. They reproduce a lot better in those conditions. And unlike what we talked about last time, 2 weeks ago in Apiculture Online, where the bees regulate the temperature very well, they don't regulate the relative humidity very well. So they can't differentially dry out the colony and keep it at a low humidity, which then would be better to control the varroa mites and other things. And so really you as a beekeeper, what you can do is place your hives in direct sun, even though you might think, oh, they're going to overheat if they're, so you want to put them in the shade. There's a balance there where actually if you have full sun or full shade, you'd probably want to choose the full sun and just make sure that they have a good water source. The ideal is to have kind of full sun in the morning and then shade during the real heat of the afternoon. But you don't always have control over those kind of things. But it's just, again, something to think about to keep that humidity down. But again, having that water source is really, really important. We talked about that last time, 2 weeks ago, about providing a water source as close to the colonies as possible. I mean, it's still a flight distance away, but not right on their doorstep necessarily. But something where it has rocks or some sort of flotation device or a drip board like this so that the bees can go on the shore and have themselves a drink of water so that they don't drown themselves. But this time of year, we're getting a lot of calls, and we always do this time of year, getting calls from people who own swimming pools. And so if you have a swimming pool, or especially if your neighbor has a swimming pool and they're not thrilled about, having bees around, this can become a real point of contention. And so, the chlorinated water, especially, I mean, we really smell it. It's incredibly odoriferous, right? Well, just imagine how attractive and how easy it is for honeybees to locate that, because their sense of smell is twice that of dogs, right? So they can be very attracted to chlorinated water, and also because they like sometimes the salts that's in the chlorinated water as well, not just attracted from the smell. And then once the bees kind of lock in on a swimming pool, it's really, really hard to dissuade them from going to that anymore. And so we'll talk a little bit very briefly about some of the tricks and things that you need to do if you have such an encounter like this. If you have a neighbor and they see your hives and they have bees coming and visiting their pool and they don't like it, these are some of the things that you can try. First and foremost, and this actually is probably 50+% of the calls that we get of, hey, I got bees at my pool. We say, well, are they really honeybees or are they something else? And more often than not, it's actually yellow jackets or something else, a non-honeybee, right? So the very first thing to do with your neighbor or somebody who's concerned is verify that they really are honeybees. If they're yellow jackets or something like that, then clearly it's not your fault as a beekeeper, and there's not much that you can do on your neighbor's behalf. Another thing that we often get is we say that, oh, the bees are swarming around my pool, right? And so, to some people in the general public, 2 or more bees flying around at the same time is a swarm. To us, of course, swarm, is very, very different. And so if you're talking about, a dozen or a few bees, it's really not that big of a deal, and they should realize that. But if you're talking about, hundreds of bees really, foraging heavily on their pool water source, then yeah, probably something needs to be done. A third thing to do is also verify that they're your bees. This can often be really, really difficult to convince somebody. They see your hives, they see bees in their pool, but it is possible that the bees are coming from somewhere else and they're not yours. And so one of the things that you can do if you really want to do some sleuthing is you can take, some nail polish or a queen marking pen or, something. And as bees are getting the water at the water source, you can paint mark their thorax with some of this paint, and then have somebody else or go back and check your hives and see if you have any paint marked bees returning to your hive. That would be proof positive, right? That those are your bees, and therefore, you want to work with your neighbor. If you don't see any of those bees returning, then it's very possible that they're coming from somewhere else. So you can do a little sleuthing that way too. Assuming that it is your bees that's going to the pool, you want to provide some other alternatives. But usually if you haven't already provided that water, once they've locked into the pool and you provide them with an alternative water source that's closer or anything else, then they'll be like, great, we have 2 water sources now, and they'll still be going to the pool. So once they kind of lock in on it, it can be pretty hard to dissuade them. So prevention is the best cure here, where providing them a water source before they find the pool is the best thing to do. However, if they really are locked in on that swimming pool, one thing that you can do is temporarily relocate them to another location for about 4 or 5 days, maybe a week, something like that, so that those water foragers that have learned where the swimming pool were will be in a new source to say, okay, that resource for water is no longer available. We're going to have to learn something else. When you move them back, hopefully they'll lock in on that closer water source that you provide them, and they won't find the pool again. So again, it's not a perfect strategy. It's not necessarily always possible to dissuade them from that pool, but those are some of the things that you can do if you have a concerned neighbor. Another thing to think about, again, as beekeepers, we definitely want to be thinking 1 to 2 brood cycles ahead so that we can prepare our bees for success, 3 to 6 weeks from now. And so next time, 2 weeks from now on Apiculture Online, we're going to be talking about sampling and monitoring for varroa mites. This is the height of the season. This is what every beekeeper should be doing right now, and really preoccupying themselves, thinking about ways to control for mites and monitoring them to see what the success of that control might be. If you have to use some sort of treatment or some sort of chemical intervention to knock those mite populations down, That doesn't mean you're done. You have to do follow-up monitoring because you want to make sure that those mites don't bounce back. You want to make sure that you know that it worked, and you want to keep monitoring to make sure that the mites don't resurge very, very quickly, because they can get knocked down and then come right back up sometimes very, very rapidly. So that's something to be thinking about for the month of August and September for sure. And so with that, I'm going to turn it over again to Dr. Ishmael Amiri, who is a postdoctoral fellow, NRC postdoctoral fellow at UNC Greensboro. And he's been doing some really, really interesting work on, well, many, many different things. One of his specialties and his expertise is in viruses, and how he showed some of the best evidence of how viruses can be transmitted sexually from a drone to a queen. But he's done a lot of other really, really interesting studies while he's here, and he's going to share with us some of his work about variation in eggs and egg sizes and why that's important to us as beekeepers. So Ishmael, go ahead and take it away.
Oh, thank you, David. And thank you for everybody for attending this meeting tonight. Yeah, I'm going to talk about the honeybee eggs tonight. And you know that honeybee egg is one very important stage of the honeybee development, because one egg can develop to a queen, or in the same time, or one egg can turn to be a worker. It depends on the amount or the quantity and quality of the food that an egg is receiving. So you see the potential of the egg that can be either worker that only lives on for a few weeks, or can turn to be a queen and living for few years. And we know that queen is laying eggs, and egg is intermediate stage of and physiological connection between queen and worker bees. And the important thing is eggs are produced by queens but taken care by worker bees. And that's very interesting, that how these two caste, workers and queens, can work together to produce a new bee in the colony. At the same time, queen who is producing all the eggs may be infected by viruses, and these viruses can pass to the next generation. And egg is playing a driver to drive the viruses from queen to next generation. But in the same time, because queen is putting so much proteins and genetic materials into the eggs, so it's possible that queen is when she is facing with all this stress and viruses, she can load more materials to the egg and make the next generation immune. But our knowledge by now is very limited about that. In a normal colony, you should see one egg in each cell. But me looking at like more than a couple of thousand eggs in the cells, I have seen many times 2 eggs in one cell or 3 eggs in one cell, and sometimes couple of eggs in one cell. They are not worker eggs, they are queen eggs. And many times I see them, queen is staying in the same place with the same cell, and she is laying eggs. I don't know what is the reason, but this is interesting to know that always it's biology always is not the same as we are thinking, that in each cell queen needs to lay eggs. And sometimes when you look back at the same frame and same cell, you see 2 young larvae are hatching from 2 eggs, and then even they go up to like 2 or 3 days after hatching. But finally, either both of them are removing from the cell, or one is removing from the cell, and the other is continuing to grow. When you look at the eggs, you see eggs are in different shape, different size. And that was like, when I look at the eggs at the beginning, where I did not, at the beginning of my study, I noticed that eggs from different queens having different shape and different size. And it was kind of question for me, what is causing different size and shape of the eggs for each queen? And that's why it comes to the question that, because we know with all the traits, genetic is the main driver, but environment can cause changes as well. And that's why we wanted to test the egg size from both sides, genetic and environment side. And we knew that there has been less investigation on honeybee eggs since recently. And that's why we started this project. And we came with a question to understand the effect of genotype for each egg, the effect of colony size, and then finally, the effect of nutrition. Genotypes, we know it for genetic, and effect of colony size and nutrition, we wanted to test it on the base of environment. And that's why in the beginning of experiment, we bought queens from 6 honeybee queen providers from different regions of the US. And we wanted to know if we keep all these queens in one apiary under the same condition, under the same environment, what is the egg size from each queen from different line, let's say line from different parts of the US. So we, in this experiment, after establishing the nukes, we let the queens lay eggs for a few weeks, and then finally we caged the queens for 15 hours during the night. And then day after, we took 20 eggs and took a picture as you see here, and finally analyzed the picture of each egg, because as I showed, each egg is having different shape, different size, and different size. And the previous studies normally were like measuring the height, weight of the eggs, but we wanted to have the whole picture of eggs. That's why we took a picture. And then using the software ImageJ, we wanted to know what is the area of each egg. So the experiment showed us that queens from different lines are laying different size of eggs. For example, queens that we bought from California are laying the big size of the eggs. Or those that we bought from Hawaiian beekeeper, they are showing the smallest size of the eggs. So, and then the other lines are showing in between of these two extreme. So the understanding is here is that different genotypes are laying different size of the eggs. Then we turn to environment, and we wanted to see if we keep the queens in different environment. Let's say keep them in nuke, mating nuke with like, let's say 500 worker bees, or keep them in 5-frame-ish nuke with maybe around 4,000, 5,000 bees, and keep them in big colony, like one deep and one super. I mean, I didn't show it here, but let's say around like 10 to 15,000 worker bees in each colony. We wanted to see under these conditions how the queen react to the environment, to colony size. After investigating of this environment, we noticed queens in the small colonies are laying the biggest size of the eggs, and then queens in the big colonies are laying a small size of the eggs. And then interestingly, the queens in the medium size of the colony are showing in between of small and big colonies. So it is interesting, and that's why we started just to continue and confirm if the size of colony has effect, or the number of bees that are available in the colony having effect on the queen, and they are and the queen is sensing how many bees are in the colony, how much food is available in the colony, how much space she needs to fill in with the eggs, and all these things. And she is like monitoring the colony and adjusting her egg size based on that. So we continue with the experiment to understand the exact role of the size of each colony. That's why we had queens in the colony, big colonies, and we wanted to see if we have the queens in the big colony and then move them to the small colony, what is the change? So we kept them in the colony, as you see here. We kept them in big colonies. We kept the queens in the big colonies, and then after adjustment for a few weeks, we start collecting eggs week after week. And for 3 times, we kept the queens in the colony. We started collecting eggs from the colony. As you see here, we had 5 queens, 6 queens that were with the big colonies. And as you see, they have, there is a variation among the queens in the same size colonies. So we collected eggs, and then we moved the same queens to the small colonies. And we noticed after moving colonies from large colonies to small colonies, there's a huge variation between the size of among the size of the eggs from 2 big groups. But still, this is how we control the environment, from big colonies to small colonies. But as you see here, there is a variation from week to week. We have variation, and that is the environment, because all of this experiment was done in the apiary, in the natural environment. And we were not able to control everything in the environment, and that's why we don't know what caused the variation. Like at the beginning, what I said, there is a variation about genetic, and that's why you see even queens, all sister queens that are in the colonies with the same size. There is a variation among them. But then from week to week, the environment that we do not have control, they cause this variation. For example, this colony number, let's say K3, I assume it is, or no, Z9, this colony. When we moved her to the small colonies, at the beginning, she was following the same pattern as the rest of the colonies. But what did happen to her, for the next 2 weeks, she started laying smaller eggs than the rest of the other queens. So beside the colony size that we had control over, there is other environmental effect that we did not have control over that, and that caused variation as well. So then we decided to test it again to confirm that this colony size has effect on the egg size. And that's why we had 12 sister queens, all produced from the same mother queen, and we had them in small and big colonies. And we wanted to see how that affected the egg size. All sister queens, 6 of them went to the big colonies and 6 of them went to the small colonies. And as you see again in the big picture in the left-hand side, again, eggs from the queens in the large colonies are smaller than eggs from the queens in the small colonies. When we look at the careful 2 groups, we see that in general, both groups are following the same pattern. The egg size is somewhere big and small. We see the differentiation between eggs from the queens in the big and the small colonies. And during the month of August, they decreased the size a little bit, both groups. And then sometimes, again, because we collected eggs in September 11th, 2018, both groups are showing increase of size. So we control the colony size and we see the 2 differentiation level, but environment has effect as well. So you see all these three experiments that I showed you, effect of colony size has effect on the egg. And then we wanted to test the nutrition status of the colony, on the effect of nutrition status on queens, on the egg size. So we again produced 12 sister queens, and we divide them to two groups. One group, we had the pollen trap in front of the colony, and then in the group, we fed them with sugar syrup. So for several weeks we did that, and every time with pollen restriction, you know, every time that you restrict the pollen from the colony, the number of the worker bees are decreasing. And that's why every time you need to, with this experiment, we added some worker bees to these colonies. We wanted to keep them in the same level of population. So we added like worker bees or capped brood in the colony to have the same almost same number of worker bees. And then after analyzing the eggs, at the beginning that you see the experiment, even queens in the overfed colonies were bigger, but slowly throughout the experiment, we noticed that eggs from pollen-restricted colonies started to increase the size, and they increased the size, but then the queens with overfed colonies, they keep the similar size of the eggs, or a little bit different, but almost similar, because statistically they are not different. So this is how the environment affect on the queen size, the egg size. What does it mean exactly? We followed the experiment a bit. We caged all the queens in the same night, and then day after, we moved all the eggs. What we did, we caged the queens and then count the number of eggs from each queen. And then we let the eggs to hatch in their own colonies. And then after hatching, and every time we were like counting how many eggs we have, how many hatched larvae we have, and so on and so forth. And then after they hatch, we move them to the common environment in a big colony with the high population of worker bees, because we wanted to see what is the effect of this large and small egg in the faith of worker bees. After we moved them to the common colony, again every step we went back and count how many of them are growing and how many of them are removing, how many of them pass to the next stage of development, how many of them become pupae and they are capped. And then later on, we moved all the frames to the incubator, have them in the cage, and for 3 days, we were counting every day how many bees are emerging from each comb belong to each queen from these two groups. And what does experiment showed us that large eggs are in the pupae stage and adult stage, or in the pupae they survive better. In the adult stage they emerge more than the small eggs. So this is one preliminary result that we got, but still the experiment is ongoing. Hopefully we have a colleague or a research visitor from China, and he is working with me this year, Dr. Han Bin, and we are working toward understanding what is the growth rate and survivability of eggs from big eggs and small eggs, and then check them. And that's why you see this egg in the upper level is egg from one day and then moving to the second day and the third day. And you see the difference between day 1 and day 2, that all the materials are started to come to the middle. And it is completely clear in the third day that materials are moving toward the middle of the egg, and you see become more visible in the two ends. And then hatching larvae. And then we are moving toward this experiment to going up to capping and understanding what does exactly queen is putting in the eggs while she is in the small colony or in the big colony. Because in the small colony, queen has only few worker bees, up to 500. And these few worker bees maybe has no time to, or no enough resources to collect and spend time with the eggs and with the larvae. Maybe queen sensed that situation, and that's why she puts more protein and genetic material for these eggs to develop in a way that they need less energy from worker bees later. But in the big colonies, first of all, the big colonies needs more worker bees, and that's why queen doesn't have enough. She doesn't have enough time to spend and load more material to the eggs, because she needs to produce more and more. And maybe because in the big colonies there are many worker bees, these worker bees are spending more time to develop and helping the queen raising all these eggs and so on and so on. So we are working on this project to understand better of what exactly queen is putting to each egg for the next step, either better development or healthier or booster or whatever. We are working on this project. So if I want to just come as a conclusion, I can say significant differences between size of the egg from different stocks, that we genetically from different stocks, queens are laying big and small eggs. Eggs from the queens in big colonies are smaller than queens in small colonies. And results suggest that egg size is actively controlled by the queen. So it seems our understanding for now is showing that queen actively monitoring the colony, what type of pollen or nectar is coming, how much is coming, how many times she is in contact with worker bees, and she is understanding the environment of the colony, and she can sense that, and she can adjust the size of the eggs based on the colony activities. And then offspring of the queens that produce large eggs survive more than offspring from the queens that produce small eggs. By that, I would like to thank my supervisors here in NC State and UNCG, Dr. Olav Rupa and David Tarpey, and Dr. Strands from Army Research Office, plus students that helped me, Carlos and Kevin, and also Dr. Hanbin that helped me this year, or is still helping me this year, and all my lab members at UNCG Honeybee Lab. So by that, thank you so much, and I'm open to any question if you have.
Yeah, that's great, Ishmael. Thanks so much. And we do have several questions from our YouTube stream, so I'll definitely get to those. But before I do want to point out just how interesting this is, and this is a fantastic example of things that are going on right under our very noses as beekeepers, and you would never really see size differences in eggs, right?
Yeah,
you
exactly.
don't necessarily even think as a beekeeper that this may be important, but this could actually be a very critical thing, right? That is both, genetically controlled, but also very much environmentally. And so when we're feeding bees or colonies different things, it could have an effect on this that then has ramifications 1 or 2 brood cycles down the line, and we're not really sure what that is. But you did answer one of the questions, or at least tentatively, you're still working on it, about does a bigger egg make a better or healthier bee? And so we're showing that they do survive a little bit better, but can you speculate on what other things that you think might be a consequence of when they finally grow up?
What we are working right now is, first of all, we are repeating the survivability of the eggs from big and small eggs, because here we had a small data set, but now we are taking it very serious and having it more control. And then in the same time, we are working on the growth rates. So, and to understand that how is the growth rate, we are checking it in a small and big colonies, in their own colonies, but in the same time, we are moving the eggs in one common colony to see other these small and big eggs in the common situation. Are they growing similar or different, or they're what exactly? And then lifespan of the worker bees, it's the next level that we would like to know if bees hatching or emerging from, let's say emerging, bees that are emerging from small eggs or bees that are emerging from big eggs, are they live longer or shorter? This is what we finally would like, because if you, let's say bees from the big eggs are living 1 day or 2 days more than bees from the small eggs, if you think about the whole colony with 15 or 20 or 60,000 bees in one colony, let's say how long it would be, and how
one day is
help
like
the trends out.
5 or 10% of their lifespan, right? So that could have
Exactly.
a really amazing impact. Another question here too, we had this question last time, but somebody asked if the workers are actually able to move eggs from one cell to another. Have you seen any of that in your work?
Oh no, not really. I haven't seen. I have worked, like I have collected and I have checked, let's say more than 20,000 eggs by now for 2 or 3 years.
Yeah.
And I have seen different size, different shape, different whatever you think, but I haven't seen, and yeah, you are right that even somebody asked last time,
Yeah.
I mean, this is biology. I said, let's say this is biology. Many things can happen, but how often? That's the question.
Yeah, never say never, but it's probably lightning strike or even more unlikely, right? So
yeah.
Let's go ahead and introduce your advisor. Olav Ruppel has been waiting patiently. He's clearly very much involved in this work and has been at UNC Greensboro for the last 17 years. And I know that because we started the same year, Olav and I did. And we've been collaborators and very close colleagues ever since. So we're really fortunate to have him. Olav, thanks for joining us.
Well, thank you for having me here.
No problem. So what is your take? You're one of the world's experts on looking at the genetics of honeybees and especially the genetic environment interactions. And Ishmael's work really kind of typifies that. What's your take on it for the beekeepers in the audience, and how do you see these genetic and environmental trade-offs?
Yeah, well, I mean, the research avenue was really interesting for me for multiple reasons. The first reason, I have to say, is self-criticizing. I didn't believe Ishmael when he first told me that there are big egg size differences.
Aren't all best studies going counter to the advisor's thoughts, right?
Yeah, well, and that's how science works a little bit. I just said, well, that's a great idea, but bring me proof, right? And so that, I think, is, the unexpected results sometimes is more exciting than the expected results. And so that's one area or one aspect of the study that really now is sort of exciting, because even after I believe that there are differences, the direction that Ishmael very quickly showed us here were in the opposite direction that I would have predicted them. I would have said, well, if you give me a large colony and a small colony and eggs in the large colony are different from the small colony, I would say the large colony has larger eggs. but it's exactly opposite. So, and we have some explanations, and as Mel explained some of these explanations pretty well. It's important to note that a lot of this variation is independent of the number of eggs that are laid, right? You could say, well, in the small colonies, there are just fewer eggs being laid, so the queen has more interest in or more resources for every single egg. But Ishmael also measured the number of eggs and didn't show that there were very much differences between those numbers, but there are differences in the sizes. So that was kind of interesting for me. I'm generally interested in this question about how complex phenotypes arise from the interactions between the genome or the genes in the bee and the environment, so that we find here a combination of factors is exciting again. Colony internal factors, colony external factors, and the genes make a difference. So that's certainly the typical scenario for a complex trait. And if we think about what we learned in school, maybe about genetics, that comes all from very simple traits. And then if we look around our environment and what we are interested in, those traits are all complex. And so it's really important to gain insights on how these complex traits are coming about. And I think we have seen over the last 10 or 20 years a revolution of our understanding of how these complex traits are inherited and how they are determined. And that's my take on honeybees. So I have to say, I'm sorry. I mean, I do have a large research program to help the health of the honeybees, but I also abuse honeybees as models for learning something more general about life and how life works. I'm a basic scientist as well as an applied scientist. And so I think honeybees are a really interesting model system to understand this question about how the phenotype or how the things, the traits that we're interested in come about, not only to help us with breeding efforts, but also just to understand how nature works around us.
Yeah, in fact, there was a question, and Ishmael, you can jump in here too and answer this about if you think that there's a latitudinal cline or an effect on this, so that if you take the same genotype and the same size, the same queen and the same size colony, but have it in the south versus the north, do you think that there's just the effect of weather or just the abiotic conditions would have an effect on egg size and some of these other things that we normally can't see?
Actually, it is like that. It's even there is an old study that they measured the egg size from, I assume it's one colony, all over the season. And they noticed from season to season, week to week, and month to month, the size of the egg is different. And it's because bees are bringing different pollen. Bees are bringing different resources. And queen is monitoring these resources that are coming to the colony and adjusting her egg size based on that. Of course, when you move colony from one place to another place. And the good thing about our study was we had all the colonies in one yard. Yep. And then we measured the egg size in two days, not like some of the colonies tonight, some of the colonies night after, or day after, not like a week between the measurement. So that's why we controlled whatever we could to show that it's not only genetic, but environment is playing. And as I showed in one of the graphs, from week to week, you see the egg size is changing. And that's why that was not under our control. But as you see, the pattern of the movement among the queens in one yard is the same. They are either going up or going down. So it is something in with the other factors in the environment that was not under our control, but they are playing a role as well.
Yeah, something communal to all of the colonies. So yeah,
exactly.
affected weather. So
And that's a very good question, David,
so-
if I can just, I mean,
Please do, yes.
here. I mean, there are general rules, biogeographic rules, right, that actually explain sort of these trends over geographic areas, typically that bees or other organisms grow bigger towards the poles, and that they're sort of close, smallest.
End up in altitude.
Yeah.
In altitude, yeah.
And along altitudinal gradients as well. So that's, that's called Berkman's rule.
That's right.
And again, I would have been not surprised to not find that any influence, because the temperature and other factors in the colony are so tightly regulated, we think, that they are regulated. But maybe we underestimated how much the external environment really goes inside the hive and influences processes in the hive.
But let me add one more thing, David, that still genetic is driving.
Sure.
Like we have, even in the sister queens, we had queens that are laying small eggs or big eggs. Either they during their development, either they were in the center of the strip that they were in the colony, or in the corners. We didn't mark that. And then having queens from different line, who knows, unconsciously, because people are selecting toward different traits.
Yeah.
If unconsciously they select against egg size, who knows that? So this is something that needs to be investigated.
Yeah. So you could be selecting for mite tolerance, but inadvertently selecting against egg size. We don't know that, but those are some of the things of why this research is really important. Before we turn it over to just some of the general questions in the Q&A, so those of you on the YouTube, go ahead and type in your general questions. But I'd be remiss to let Olav off the hook without talking about some of his other research. He's not only one of the world's experts in honeybee genetics, but he's also a leading expert in the study of aging using honeybees as a system, because there's such a great system where you have the same genetic type that can become a long-lived queen or a very short-lived worker, and then everything in between. And so, what new studies do you have going on, Olav, and make the argument as to why honeybees are a really great system to study aging, and that might even have implications for us as humans.
Yeah, that's another good example why we can use honeybees for wider biological questions. And I would say that the research project that you heard about today is separate or from my interest in aging, because ultimately these processes of development and then how organisms live their life and finish their life or age at the end might very well have some connections. So again, we don't know that for the egg size study yet, but what we see in general biology is these very long-lasting effects, sometimes even across generations, that affect the health and well-being in old age. And that's an interest of mine, aging. We all age, right? And it's an almost universal biological feature. But honeybees are special. I mean, they are special in our hearts, but they're also special because they live in these social evolution societies. And that has led to some exceptions to biological rules. So one of the rules about aging is that you can either reproduce a lot and sort of die quickly, or you reproduce slowly and less, and then typically you live longer. And those trends, those rules are applicable when we look between different biological species. So if we, let's say, compare a bunny with a elephant, okay? But they also apply to within species. So if we are, let's say, comparing humans and their number of offspring or children and compare that to their average lifespan, not in modern societies, but in prehistoric societies, that is very tightly correlated. And so there's something that is called the cost of reproduction. The more you reproduce, the shorter you're going to live, and that's sort of a cost, a physiological cost. That is one of the many sort of examples of life history trade-offs. Nothing is for free, basically. Right?
And here we just thought it was having to put up with teenagers.
Yeah, right. And honeybees are different, of course. All of you know this. Honeybee queens reproduce 2,000 eggs per day, maximally at least, and they still outlive their workers for a long, long time. Yep. And so that's a very exceptional observation. And when you have an exceptional observation, that warrants investigation. And so, of course, we can compare workers and queens, and a lot of studies have done this, including a few of ours. But I think the better way to study what really causes the lifespan extensions of queens is to separate the effect of reproduction from anything else that separates a worker from a queen. And so one of our studies has looked at laying workers. And these are workers, of course, that are switching on their reproductive system, that are acting a little bit like queens, but are clearly not queens. And so we're trying to see what confers longer life to these laying workers, because they do live a lot longer than regular workers. In one of the studies, we hit them with a lot of viruses, and we hit them with paraquat, which is an oxidative stressor. And it turns out that these reproductive workers are more resistant to both the paraquat, which is actually a herbicide, and Israeli acute paralysis virus. So this reproductive activation seems to strengthen their immune system. It seems to strengthen their defenses against stressors. And that's very different from other organisms. Again, in other organisms, typically, the more resources get put into reproduction, the less the organism has left over for defense of its own body.
And again, finding out the mechanisms that govern that is really, really important, because if we can just tap into that a little bit, again, a 1 day, survival difference of the workers can be like 5% of their lifespan and tremendously help colony health and productivity, right? So
I wouldn't recommend to dequeen all your colonies.
No. Don't turn them into laying workers. That's right.
Don't do that. That's not a good idea. But and to be honest, these molecular signaling pathways that we're finding and the genes that are involved in these lifespan extensions, they are highly conserved. So there are genes that are doing the same thing in our bodies. Obviously, they're also doing different things because we have not this positive effect of reproduction on our aging patterns. But if we can understand it better in bees, maybe we'll understand it better in humans as well.
Yeah, no, that's great. Well, there's lots of questions flying in. There's a lot of questions here, Ishmael, that you might want to check and see, but I'm going to answer all of them in the same answer, which is we don't know yet, and Ishmael needs to follow up on it, such as, do larger eggs produce larger workers? Is there evidence that effect between egg size and varroa mites? Would a larger egg be more for the, varroa mite to feed on? Is there effect on division of labor? Does treating the hive for varroa have an effect on egg size? We don't know any of those things, but again, that's hopefully something within this paradigm that we might be able to see. There was a question earlier on, kind of a very general question, because we only have 5 minutes left, so I'm just going to open it up to everybody here to chime in. There's one really good question about, because it is kind of varroa mite treating season, what to do and whether or not you're going to leave honey supers on if you're going to treat for varroa mites. And again, others can jump in here if you want, but in general, it depends on what you're using. But in general, you don't want to have honey supers on when you're treating for mites, even if you're not planning on extracting that honey. What we like to do is have the deep frames, and any honey that's in the deep frames is what you keep for the bees, and you don't have to worry about, any treatments that go on. But we have our honey supers in shallow frames or medium frames, so that we know that we never have any of those types of boxes on our colonies if there's any kind of miticide treatment in there. And so even if you're not going to harvest it this year, you might forget, and it might get mixed up, within your operation later on. So it's usually a pretty good idea, unless you're really meticulous about, keeping frames separate and labeling them and stuff, to take those honey supers off while those treatments are in. Folks from the NCDA, would you guys agree with that advice?
Yeah.
Okay, Don, man of many words, but I'm glad to hear that. Good advice, I think. All right, so let's see some other questions here. Who wants to take one about? I saw it here before. Oh, here's one that kind of talks about what Olaf was just talking about, laying workers. Can laying workers go on mating flights? I think the answer, that is, the answer is no, thank goodness, right? Otherwise they'd take over.
Yeah,
how long do laying workers live? That'll get back to your question about the aging. How long do they live?
And so, the whole, how long does anything live is a really complicated question, because it's an individual property. Every individual has a certain lifespan and dies at a certain time. But it's also a population property, right? So we can say on average they live, 100 days or so. But it's a complex trait, as we said before. So it's influenced by a lot of different things. It might depend on the genotypes of those bees. It might also depend on the season, obviously, because the colony is eventually doomed. So in the fall, these reproductive workers wouldn't have as long as a lifespan as in the spring, when the colony becomes queenless. And it depends on a lot of other social factors as well. They are certainly not living multiple years, but queens don't do that anymore either. At least not very many of the queens.
they used to, but that's another kind of bucket of questions, right? Of trying to look at longevity within the queens and not just the workers.
Yeah.
Well, I think just for the sake of time, I'm going to end at the top of the hour. Really appreciate everybody's questions. I do want to remind everybody that we are doing these every other week. So our next Apiculture Online is going to be August 12th. Again, starting at 7 o'clock, live streaming on YouTube. Make sure you like, comment, and subscribe to our channel. That can keep this going as long as we can, as long as there's interest. Again, because the timely topic is the height of the varroa mite season, and so we'll be going over how to sample for varroa mites. And we're going to have a special guest from the USDA, a lab alum, as well as a co-advisor with Dr. Ruppel, Dr. Mike Simone Finstrom, who is doing a lot of work on the interactions of varroa mites and viruses and many other things in their genetics and bee breeding lab down there in Baton Rouge. And so be sure to sign up for our YouTube channel and subscribe to our YouTube channel. You get automatic notification of our next Apiculture Online. Until then, thank you again, Olav. Great job again, Ishmael, on presenting the timely topic and your research. We look forward to more great things to come from there. And thanks again for everybody attending.